Hello everyone and welcome to week 6, and my name is Catherine and it's about 1:30 on June 11th, and I'm going to be talking about the environmental influences on microbial growth and the salt tolerance testing. Okay, but first exercise we're doing today is on the tolerance. We're going to be using the nutrient BR with 1 NaC milliliters, 1% for the epidermidis, sorry, and 7%, and then the 15% along with 1% cerveza and 7% and 15%. And I labeled them all here. I have my alcohol ready. I have all of the actual bacterias, and then I have my bleach here ready. And I'm going to go over a few things first. So I'm going to talk about the environmental conditions affecting microbial growth, and it's the solutes water activity, like the pH, temperature, oxygen level, pressure, and radiation. Okay, then the osmolarity, concentration of solution, and it's the total amount of particles per liter. And then I'm also going to talk about the hollow tolerant, and it's where species tend to live in the hypersaline lines, like the dunes or the salt deserts, the inland salt, and the springs. Also the halophile, and that's like the microorganisms that growth of where it can be in the saline conditions. And then where microbes occur is conditions where bacteria enter in the body and populate, or the cells in the body are damaged, like an infection, So those are the type of the type of environmental areas of which the conditions for microbial. Okay, and I'm continuing on now. Gonna start. So I have my S. epidermis. I'm gonna do this quickly, and I need to go ahead and open them both. So leaving one here, one lid here, and I'm gonna go ahead and do like I always do. We're going to sterilize the tip. Okay. And you just need to run it through the flame just for a little bit. And then I'm going to take the one that has the broth. Okay, and that only needs 2 drops. So I'm going to try to do this as quickly as possible. So remember, this one is the broth that's not labeled, the 1%. So you want to make sure this is dry. So I'm only going to need to put 2 drops. So I'm going to transfer 1, 2. Okay, and I'm going to put it back in here. Okay, but I'm going to sterilize the tips on the top of both. Oops, I'm going to go a little bit too close. Okay, you kind of know what the idea is here. I can actually leave this one open because I'm going to be using it. But this one I'm going to put back on. Okay. And I'm going to do it again with the 7%. That lets me open it. And since I already, I sterilized that, I probably will do it again only because it's been in the air for a few minutes. So I'll go ahead and do that one again. And then just making sure that the alcohol is dry from inside the pipette. And I'm going to take 2 drops from the one that's not, oops, still has some, okay. Just kind of be careful of so I'm going to put 2 drops of 1, 2, okay. And again, sterilize the tip of the 7%. Okay, these ones are much easier to stand up because they're not so hard to think the other ones were more of a glass. These are more of a plastic. So I'm going to do the same thing, sterilizing the tip. Sterilizing the tip. Okay, doing the same thing. It's kind of, you get familiar after doing this so many times. So, and again, I'm only going to take 2 drops from the broth of the Epidermidis. Okay. So 1, 2, okay. And then, you don't want to do that. What am I doing? Okay. So I need to go ahead and sterilize the top. And this one is actually the culture. So I'm done with this one. And I'm gonna do the 15% as well. And I'm wanting to remember to do the most important part is to move the vials back and forth after each one, because it actually mixes together. Okay, so next I have the S. cerevisiae. So I'm going to go ahead and sterilize the top of this one. Okay, and then here we go. I'm using the 1%. I already have it labeled. Okay, same thing. It's easier. Okay, so now I'm going to use another pipette. Okay, and I'm only needing to take 2 drops. So this is the one that's 1%, and this is the one that's the broth. So I'm going to take 2 drops from here. So here's 1, 2. Okay. It just helps to clean it out a little bit. Okay, so I'm going to go ahead and sterilize this one again. This is the 1%. The lid back on. Okay, this is the actual vial of the bacteria. Now we're on the 7% of the S. cerevisiae. And this again is exercise 1, if you're not familiar. Okay. And then okay, so you want to make sure it's dry. And I'm going to take it from this one, because this is the actual bacteria. And you only need 2 drops, so I'm trying to not waste a lot of it. So 1, 2, okay. Sterilizing this again is the 7% of the S. cerevisiae. And this is the bacteria. Okay, go ahead and cap this one. Moving on to the last one, that's 15%. Sterilizing this one. Oops, that's a scary one. Okay, and then the top of this again. Okay, and this one's pretty self-explanatory. You have to do like a 48-hour incubation. So just to remember. Okay, so you're going to put 2 drops of here. So inside here, 1, 2. Okay, and then we'll sterilize that. Okay, now let me go ahead and do this one. Okay, so that's pretty much done. So again, this is the 15%. And of course I didn't move it around as much as I should have. Okay. And let me go ahead and sterilize this. Now this is going to be again a 2-day sterilization. So I will give you the results in the next 48 hours. Until then, I hope you enjoyed this video. Thank you.